Hi class and welcome to lab 4. This lab is on microscopes and microscopic cells, and your worksheet will involve labeling a microscope and then doing a couple of lab simulations on different websites, looking at microscopes and seeing things underneath the microscope. So here are the different types of microscopes: the compound microscope, the phase contrast microscope, the dissection microscope, the scanning electron microscope, and the transmission electron microscope. So first of all, the compound microscopes, these are light illuminated. So light will shine up through this part right here, and it will shine up through a specimen or a sample. For example, you can see two samples here, and then you look through these ocular lenses at the sample. The image seen with this type of microscope is two-dimensional. The microscope is the most commonly used. It's the microscopes that we would be using in lab if we were in person. However, it has extremely low resolution, so it's not very clear, and it cannot get as zoomed in as other microscopes can. This is an example of a human blood sample. The red or pink little circles are red blood cells, and anything stained purple is a white blood cell. And here's a look at a microscopic view of a plant stem. The phase contrast microscope is used to enhance the contrast of light microscopy images of transparent and colorless specimens. So oftentimes, if we look at something, for example, in the light microscope, we can't see the contrast of structures within a cell. So what the phase contrast microscope does is it enables the visualization of cells and what's inside of cells that would be more difficult to see using an ordinary light microscope. So it just provides some sort of contrast between the details. And this is a look at an amoeba. The dissection microscope is also light illuminated, so a light shines down on top of the specimen. The image that appears is three-dimensional. It's used for dissection and to get a better look at a larger specimen. You cannot see individual cells because it has a very low magnification, so you would actually put maybe an insect or a leaf underneath the dissection microscope. It has extremely low magnification, so it's a little bit zoomed in, as you can see the head of this insect here, but it's not super zoomed in. So it helps in dissecting a specimen. So that's why we give it the name the dissection microscope. The scanning electron microscope is really neat. It uses electron illumination, so it beams electrons at a specimen, and then the electrons bounce off to give an exterior view of the specimen. So the image seen is 3D. It has extremely high magnification and high resolution. The specimen is coated in gold, and that's what the electrons will bounce off of. So here's a look at the scanning electron microscope. It's a much more elaborate machine that we would not use in lab. Here's the head of a bee, so kind of the detail and the zoomed in that you can get from a scanning electron microscope. And then a blood clot. This is a really cool picture. It's showing the red blood cells and all of the fibrin and platelets kind of clotting them together to try to stop someone from bleeding. The transmission electron microscope is also electron illuminated. It gives a 2D view, so we get very thin slices of a specimen obtained. The electron beams will pass through this, and it has very high magnification and high resolution as well. The only difference between the transmission electron microscope and the scanning is that the scanning is 3D and the transmission is 2D, but they both use electrons to pass through the specimen. So this is a great slide. You guys have to fill out on your sheets and label a microscope. So if you want to pause the lecture video here or download the PowerPoint, but this is a great slide to use to do that part of your worksheet to label the microscope. You look through the eyepiece, also called ocular lenses. Here's the slide holder. So you'd put your slide with the sample right here within these clips. The objective lenses, there's usually 4 of them in the compound light microscope, and they just turn on an axle to give you a more zoomed in look at the specimen. Here's the light switch, so it turns the light on that comes up through here, this substance light. The iris diaphragm lever controls how the light travels through and up the condenser, and the condenser will condense or make the light stronger as it travels through the specimen slide. The stage control knob, so this is the stage, and it can move back and forth, right and left, and the stage control knobs will control that. And then you have a fine focus and a coarse focus knob. These help get the specimen or the sample into focus. So how the microscope works, light travels from the bottom of the scope through the specimen on the stage and through both an objective lens and the ocular lens. So the objective lens was one of four lenses that is used to magnify the specimen. And then the light also travels through the eyepiece or ocular lenses. So the image that reaches our eyes is bigger by both the magnification of the objective lens, which are these guys, and also the ocular lens or eyepiece. So to find out total magnification, or to get the total magnification power, we multiply the objective lens, which is always 10 times or 10x, and we multiply that by the difference of magnifying power of these four objective lens. So total magnification power can be seen as 40 times a specimen, so 40 times zoomed in, and that comes from 10 times in the objective lens multiplied by 4 times in the lowest powered objective lens. So 10 times 4 is 40. Another objective lens magnifies it 10 times, so 10 times 10 is 100. Another objective lens magnifies it 40 times, so 10 times 40 is 400. And the highest objective lens power magnifies it 100 times. So we take 100 times the 10 of the ocular lens and we get 1,000 times magnified. Images are always inverted in microscopes. So what that means is what you're looking at, if you were to place a specimen underneath the microscope, the image is always inverted, and that means it's backwards and upside down. So if we were to put the letter E right side up in our underneath our microscope in the sample, what we would actually see is the E upside down and in reverse. So that's what the microscope does. Here's the low power objective, covers the wider field, so it's not as zoomed in. And the high power objective gives a larger image because it zooms in on the E more. And you guys will be working with the letter E in one of your worksheets for this lab. I think it'll take you to a website and it'll just ask you to draw what the E will look like underneath the microscope. And it basically looks like this. The E is upside down and backwards, and that means inverted. In using a light microscope, the specimens often have to be stained to be seen in extremely high contrast. So this is where the color comes from in many of the images that you see. So for example, this is a look at some plant cells. Normally plant cells aren't stained this purplish bluish. This color comes from the stain that is put on the plant cells so that you're able to see the detail. Here's a look at the cytoplasm and nucleus and the cell membrane of an animal cell, an amoeba, actually, and then this is what it would look like in artistic rendition. Here's a look at a cell with cilia and all of the structures within. Here's a look at E. coli and seen under 1,000 times magnified. Here's a look at cholera. So kind of just examples of what different bacteria and infections would look like under a microscope: Giardia, Paramecium, the amoeba, the Volvox, the Elodia, and then onion skin cells. So again, showing how different stains provide different color. Onion skin cells again, the cheek epithelial cells. So we could take a sample from the inside of your cheek to get this look, and then we would stain it to put underneath the microscope so we could see that. And then the rest of the slide just gives you different links to follow to look at different organisms underneath the microscope, and I'll let you guys do those on your own. So again, this lab is due on Sunday night, as well as the quiz from the previous week's lab. So thanks for listening, guys. I hope you are all doing well.